Okay. Yeah, thank you very much, Beth. Yeah, thanks everyone for being here, even if here is virtual. I'm really excited to tell you a little more about this spatial capture recapture model for group living species. So for context, oftentimes we care as much about estimating the abundance or density of groups in ecology as we do about estimating the abundance or density of individuals. This is because group size and structure and abundance can affect everything from individual fitness to population dynamics all the way up to ecosystem processes. And for a little background on what we've done, what has been done so far for estimating abundance of group living species, there's been previous work to adapt models in capture mark recapture, distance sampling, and end mixture to account for things we might care about with group living species, counting groups and individuals, and modeling dependence and detection between individuals. There hasn't been quite as much work done in this area on spatial capture recapture. There have been some sort of assessments of the effects of these of groups and individuals and detection dependence, but not as much work. And none of them allow us to allow group ID to be inferred. They all assume group ID is known. So with FCR in particular, there's no straightforward way to model groups and individuals simultaneously. It assumes independent activity centers, which can be violated with group living species, and it assumes known group identities. Now, spatial partial identity models, or SPIMs, offer a potential solution. These use partial identity markers like sex or photos of a single flank to assign detections to individuals. But furthermore, we can also use the times and locations of detections to assign individuals to groups if we kind of expand on this model. So the model that I'll be telling you about infers group identity of individuals while accounting for dependence in detections. So the components of this group SPIM model are a double data augmentation procedure to estimate group and individual abundance, group level activity centers, and a model for group size and group site visits. Then there are individual detections that are conditional on group site visits, so an individual can't be detected at a site unless its group also has the potential to be detected there. And then finally, a group identity vector that links individuals to groups. So with that, I'm going to delve into the model. We start with group data augmentation. So we have a group inclusion parameter Z sub G for group G. That's governed by an inclusion probability psi. Then we have a group identity, gamma sub I for individual I. This can take values of 1, 2, all the way up to capital M, which is our superpopulation of groups. And so that number just sort of tells you what group an individual is in. Then we have an individual inclusion parameter, W sub I for individual I. This doesn't have a Bernoulli distribution like you might expect for traditional data augmentation. Instead, it's linked to our group size, N sub G, or which is essentially the number of individuals in group G that are included in the population currently. And that number is going to be distributed zero truncated Poisson with mean mu. So there can be no groups of size 0. Then, like traditional data augmentation, our group abundance G we get from summing the group inclusion parameters, and our individual abundance N we get from summing our individual inclusion parameters. Now moving on to some more SCR processes that might be familiar to you. We have group level activity centers, S sub G, that are distributed uniform over the state space. We have a group site visit detection rate, which is lambda sub G J K for group G trap J occasion K. That's governed by a baseline detection parameter, lambda naught, a scale parameter sigma, and the distance D between our activity centers and our traps. Then site visits V sub GJK are distributed Poisson with mean lambda sub GJK. And that's sort of the part of SCR that you all might have seen before. And then finally, there's this individual detection component, Y sub IJK for individual I. That's Poisson, where we have the individual inclusion parameter included in there so that an individual can't be detected unless it's included in the population. We have our group site visits for that individual's group, and then our baseline detection parameter, theta naught. So just to illustrate this detection process a little bit, we have a detector array here, these black X's, and then we have activity centers, in this case for 2 groups, group 1 in blue and group 2 in green. And based on distance from these activity centers, we generate group site visits, which are these squares. And then anywhere there's a group site visit, you can also have individual detections. And you could have anywhere from 0 up to potentially, right, some very large number, infinity individual detections. Now I want to tell you a little more about the MCMC algorithm we're using to implement this model. So most of our parameters, like our detection parameters, can be updated using basic metropolis tasting steps. Group identities are updated using a slightly more complicated metropolis tasting step that proposes one of 3 different things. Either moving one individual from one group to another, group fission, which is when we split individuals from one group into two, group fusion, which is when we combine individuals from two groups into one. And this is similar to birth, death, move algorithms you might see for Neyman Scott type point processes or finite mixture models. And then we also have a metropolization steps to jointly propose to switch a group on or off, and also switch off its n sub g members simultaneously. I want to give you some examples of how some of these group identity updates work. So for instance, if we want to move one individual, let's say we have two groups, 1 and 2, with group sizes 4 and 3. So that's the number of individuals that are currently switched on in that group. And all these gray dots, these gray individuals are individuals that are switched off, but still included in that group. So if you want to propose to move one individual, for instance, we can propose to take one individual from group 1 that's switched on and move it to group 2. And that updates our counts to 3 and 4 instead of 4 and 3 for groups 1 and 2 respectively. Now for group fusion, what we do is we have two groups that are both currently switched on. What we want to do here is propose to switch one of our groups off, in this case group 2, and move its switched on individuals over to group 1. So under that proposal, we switch off group 2, we move its switched on individuals over to group 1. So now the count over there is 7. We actually keep the underlying latent count of 3 for group 2, just in case we want to switch that group on again, so that we know how many individuals to include. And then we keep the gray individuals where they are for now. And then for fission, let's say we want to switch a group on. What we can do is propose to essentially steal 3 individuals, since that's our count for group 2. So we steal 3 individuals from group 1 and bring them over to group 2 and switch group 2 back on. We were lucky enough to be able to apply this to a case study of African wild dogs. So we applied this model to detections of African wild dogs collected August to November 2014 in the Okavango Delta region of Botswana. Excuse me. And this was from 132 cameras deployed for about 35 days each. And overall, we detected about 66 dogs in 6 packs and included these in the analysis. Now, what's really cool about this population is that we know individual and group density and identities, or at least we have previous estimates of individual and group density, and we do know the identities of any dogs we detect. So we use group SPIM to estimate individual density and group density and compare this to previous estimates, and then also tried to infer the group identities of detected individuals. And so we were able to use AUC to measure group identification success for these individuals. We also simulated data. So we simulated some bias correlated random walks of group movement. I should note this is a different model than the one we fit. We wanted to see how this model would work with a more realistic movement process than the data generation process. We had a fixed abundance of 250, or sorry, than the model process, excuse me. We had a fixed abundance of 250 and either 10 or 25 groups with varying group size. And then we had movement speeds that correspond to 3 different long-term home range sizes. And for each of these, we fit our group SPIM model, got our individual density, and also calculated pack AUC. So a few details on our MCMC implementation for our case study. We ran 3 chains for 50,000 iterations each, with uniform priors in all of our parameters, and a single occasion. And then for our simulation, we ran 1 chain of 3,000 iterations each, and we did check this model converges pretty quickly. So hopefully that should be actually okay in this case. We ran uniform priors in all parameters and had a single occasion. Now delving into our results, we found that group SPIM recovers wild dog density in groups fairly well. So what we're looking at here is a table of our mean and 95% credible interval for individual density, group density, and average group size from our wild dog model, compared to previous estimates for the area. So you'll see that our mean of 2.4 dogs per 100 kilometers squared is not so far off from our previous estimate of 3.5 dogs. And that 3.5, the previous estimate, also falls within our credible interval. Our group density and average group size, as you can see, were a little farther off from previous estimates, but our credible intervals for those quantities still got fairly close to the last known values. And then when we calculated AUC for group identities, we found that that averaged 0.95 across 3 chains, so not too bad. Now delving into simulations, we found that group SPIM accuracy varied by aggregation and by movement speed. What we're looking at here is our estimated abundance from our simulations on the y-axis, number of groups on the x-axis, and the red line is our true abundance of n equals 250, and our panels are our different movement speeds and home range sizes. So for 25 groups, you'll see that on average we did pretty well. We were pretty accurate in terms of abundance, and we were more biased when we had fewer groups, just 10 groups. And we also had slight differences in precision across our movement speeds, or sort of, excuse me, the variation in our simulations. Now looking at our AUC for our simulations, we found that we actually recovered group identities pretty well across the board. Nowhere did we get below 0.75 for AUC. So to conclude, we found that group SPIM estimates density well under some aggregation scenarios, and that it recovers group identities pretty well across the board, both in our simulations and case study. And then it has potential for ecological applications. Since we're modeling group size and number of groups and number of individuals, we can look at drivers of group size. We can look at, we can monitor groups and individuals over time to look for evidence of allele effects and what's driving those. And our next steps are to run some more simulation scenarios, to build models with multiple survey occasions, and then to incorporate hopefully an explicit movement process into our spin models. With that, I'd like to thank my co-authors and Sarah Converse, Devin Johnson, Nathan Hostetter, Paul Kahn, and Richard Chandler for some help with these ideas at a workshop last summer. And then the Quantitative Conservation Lab at UW and my lab, the Quantitative Ecology Lab, for their help with this talk. With that, there are references if folks are interested, and I will take questions if I have time.